to introduce today's speaker, Dr. Geoff Pegg. Geoff is a forest pathologist and is currently the Forest Production and Protection Team Leader with the Queensland Department of Agriculture and Fisheries. He has been a researcher in forest health and biosecurity since 2000. And I'm thinking now would probably be a great time to have him on screen. Apologies to anybody if this goes a bit skew. This is my first time hosting one of these webinars. And I hope you are now seeing Geoff. Right, his recent research focus has been studying the impact of myrtle rust in native Australian environments, raising awareness of the importance of environmental biosecurity and understanding the threats pests and diseases posed to Australia's ecosystems are key areas of interest. Please commence your webinar, Geoff, and I'll be back at the end to facilitate the questions.
Thanks for that, Renee. Hopefully everyone can see my screen now. So as Renee said, I'm going to present some of the work we've been doing recently, looking at the impact of myrtle rust on post-fire regeneration in Australia. So 2019-20 summer was one of our most severe fire seasons on record, with fires affecting all states. In particular, New South Wales is one of the worst hit states, with over 5.5 million hectares burnt. That included 2.7 million hectares, or 37% of the national park estate. 53% of one of the world's heritage Gondwana rainforests, and this area is one of the most important subtropical rainforests, or the largest subtropical rainforest in the world. It's of extremely high conservation value, and 53% of that area was affected by fire at some level. I'm just going to show you a few shots to give you an idea of the impacts of fire. This is in Bundjalung National Park, which is Northern Rivers of New South Wales. It's a coastal heath and woodland ecosystem. This photo was taken around January after fires went through in November. And we can start to see here the regeneration occurring both in the heathland ecosystems and the woodland. This just gives you another idea of the extent of the damage that occurred in those fires. In the top right there, you've got all the nice coastal heath, which is really myrtaceae rich environment. And in the background, we've actually got the eucalypt forests or woodlands showing significant fire damage as well. So not all areas did have intense fires. We had some areas where right through the big mature eucalypt forest, we had fires right through the top of the canopies. Other areas we saw fires just in the undergrowth. And then also we had fires in what would normally be a wetland, but because it had been so dry prior to the fires, Melaleuca wetlands burnt so easily, and we saw a significant dieback as a result of those fires in those ecosystems. So just to give you an overview of the research, as I said, we're looking at the impact of myrtle rust on forest regeneration after these wildfire events. We're looking at the incidence and severity of impact in different regions. So we had sites across different climatic regions, including the subtropical regions and the temperate zones down in southern New South Wales. We had a look at different forest types. As I mentioned, some of the coastal heathlands, the woodlands, and then some of the high altitude rainforest ecosystems. We looked also at the different fire intensities and to see whether rust was impacting on these ecosystems. So where we saw intense fires going through the canopy versus those in the understory. We've also established some plots in different sites to try and look at the progression of disease on the different species. We wanted to, first of all, obviously look at which species were susceptible. Within those species, was there variation in susceptibility and impact caused by rust? We also want to look at the types of regeneration that are affected, including seedlings and reshoots. Some species, they regenerate by seedling only or reshoot only, and other species regenerate using both seedling and reshoot. So just giving you an idea of where we conducted our studies. So we've, as I said, we've established some monitoring plots, but a lot of these sites were also done by one-off surveys through these areas, trying to cover as much area and investigate as many species as we could within these ecosystems. So in the northern rivers of New South Wales, up in the top there, we had areas of coastal heath and woodlands, eucalypt forests and inland woodland sites, the Gondwana World Heritage areas, Then the central coast of New South Wales, we also had coastal heath and woodland ecosystems in this area, littoral rainforest systems, and then the southern New South Wales down to the Victorian border, we had coastal woodland sites. We had hoped to extend into Victoria, but due to COVID restrictions, we weren't able to cover any of these sites. This just gives you an idea of some of the different sites we're trying to look at from a coastal to an inland area, and shows the mosaic of different fire intensities within these ecosystems. So on the left-hand side there, we can see that we've looked at the coastal heath areas where there's been intense fires, heading further west into some of these woodland environments, and then right up into the ranges where we've got the high altitude subtropical rainforest ecosystems. You can see on that right-hand side the really dark coloured in the image there is where the most intense fire systems, and that's that Bundjalung National Park that I showed in that first image, showing that level of impact caused by those fires. In Queensland, we selected sites. It's primarily in southeast Queensland. The first site we had a look at was on Gari, which is the World Heritage Fraser Island National Park, and that included coastal heath and woodland. But the focus really there was on Melaleuca wetlands, and in most of those areas, even though we haven't mapped it on this particular image, we did see quite intense fires going through the canopy of all the Melaleuca wetland areas. We also included the Great Sandy National Park, where we had a range of different fire intensities and also a range of different ecosystems within there, including the coastal heath and woodland and the littoral rainforest areas. And then there's right in the southeast, this is where Queensland Gondwana World Heritage areas are of the rainforest ecosystems of Lamington National, make the bunch of the main range national park. And you can see there also a range of different fire intensities, with red being the most intense. So just to give you an overview, I guess, of the results, I will actually focus on some particular species and give some more information in detail on those. So what we did find was myrtle rust impacts across all sites apart from those in southern New South Wales. And that was despite the fact that we actually saw significant levels of rainfall, fantastic new growth occurring. We did not find any evidence of rust occurring. This was, I guess, what we consider our temperate regions, but it was quite surprising that we didn't find any evidence of rust within these sites. So rust was detected pretty quickly after we had the fire regeneration occurring. In sites where we saw fire in November, we had regeneration around January, end of January, after the first lot of significant rainfall. And then we started to see rust come into these sites around March. Once they had come into these sites with everything flushing beautifully, all the different myrtaceae throwing beautiful flush after that fire event, we saw a rapid increase in disease incidence and severity. We did find disease in all fire severity levels, although there seems to be some indication that in lower fire intensity, some species haven't had the same level of rust impact as others. We've seen disease on reshoots and seedling regeneration. And when we look at the number of species we've looked at in these sites, we've seen about 36 different species of myrtaceae identified with some level of impact of rust. Within these surveys, we've actually found new species records, particularly field host records. Some of these have been identified as being susceptible in glasshouse studies. Leptospermum speciosum, Melaleuca squamea, Eucalyptus amplifolia, subspecies amplifolia, were three of the species that we've identified as new host records in the field. We've also seen impacts on range restricted species, including the Eurymyrtus australis, or the peach myrtle, which was endangered, or is listed as endangered, I should say. It currently occurs in the nightcap ranges and is restricted to that area only. The photo that's on this slide is a picture of a root sucker coming up post-fire that's covered in rust. Now, it doesn't get that typical yellow golden spore mass, but you do get significant dieback as a result of rust infection. The good level is that good news is that we don't see infection on all the sites that we've got in our study plots at this point. We've seen impact on threatened species, but we've also seen impact on common species such as Melaleuca and Eucalyptus, and I'll cover these now in more detail. So the first species I want to talk about is Melaleuca nodosa, or the prickly-leaved paperbark. It's common in coastal regions and tablelands of Queensland and New South Wales. And in the coastal heath areas, or any area closer to the coast, it grows more like a shrub. almost like a hedge like habit. But then as you head further inland, it tends to grow more like a small tree, some trees reaching up to 10 metres in height. So its regeneration post-fire is through resprout and seedling, although we haven't seen many seedlings of Melaleuca nodosa in most of our study sites, apart from a couple that we've seen infected. This just gives you an idea of the symptoms we've seen. So pretty soon after we started to see regeneration from reshoots on these particular species, we started to see infection, heavy levels of infection on both the foliage and stems. As a result of that infection, we started to see pretty significant levels of dieback on the regeneration. And you can see on the left-hand side there, that's one of the woodland tree habit. And the right-hand side there, we've got more of that coastal hedge like habit. In the middle there, we did actually capture some evidence that we were getting seedling infection, because the frequency of when we were doing the assessments in different plots, and because the one-off surveys, we really haven't got data to quantify what the impact would have been on seedling regeneration of this species. This image just shows you the differences on the top two, going from the left there. We've got individuals that are showing some tolerance or low levels of impact from rust versus those where we're seeing significant branch dieback, almost 100% occurring on the others, with all the new flush being burnt off by rust. So this is just looking at some results from the study sites we've established. This is looking at one-off surveys within the different ecosystems. Unfortunately, we don't have any Queensland sites of Melaleuca nodosa that were affected by fire, but these are sites that range from Central Coast in New South Wales up to the Northern Rivers in New South Wales. We've got a combination of heath sites and those woodland sites where we've got the small forest trees. If you look at the results from the heath areas, there is some indication that fire intensity is influencing the level of impact that rust is causing. And I've categorised the level of impact on this particular data based on the level of dieback that rust has caused, with those trees or individuals showing greater than 50% of branches with dieback seen as significant levels of impact. Again, similar sort of results of what we're seeing in the tree. The lower the fire intensity, there is some indication that we're getting a pattern there where we're getting less impact. But we do have some individual sites there, the Banyaba site, that's also showing low levels. Now that site, in particular, when we did that assessment, regeneration had only just commenced. And so we're only just starting to see rust come into these sites. So I don't doubt that when we go back to those sites, that we'll actually see some quite significant impact based on that level of impact, the level of infection we were seeing on the new flush and regeneration. As I said, we do have some monitoring plots set up within these sites, and the aim is obviously to look at the impact of repeated infection on plant survival or plant health as well. What we've seen in other sites and other species, it may take a number of years of repeated infection before we actually see tree death. Here we're just looking at a 6-month period where we've seen quite significant dieback. So what's going to happen over the next few years if we get repeated infection is something we need to understand. But what's interesting here is that you can see that the level of disease is pretty well linked to the flush events. So we get this up and down nature occurring, where you get a flush occurring, you get a high level of disease. The disease kills off all the new flush, you get no flush, you get a drop in the disease. As the flush increases, we also start to see an increase in disease occurring. Fortunately, we were able to, with one of our surveys, capture some flower data and the impact that myrtle rust might be having on flowering of Melaleuca nodosa. This is one site in the Bundjalung National Park in the coastal heath areas. And what we're seeing here, we've categorised this into impact based on the level of branch dieback with trees with less than 40% of branch dieback, and there's only 25% of those trees existing within that category. 60% of those trees had greater than 50% of the branches covered in flowers. Whereas if we compare that to trees where we had greater than 60% branch dieback, which was the majority of the population with 53% of individuals showing that level, we had most trees with less than 50% of branches showing some level of flower, and 5.7% of those with absolutely no flower at all. So whether we'll see with this repeated infection this changing, the good news might be we can suggest that we do have evidence of tolerance, and that perhaps we can easily select for tolerance within these sites for regeneration, or you might say that there'll be some natural selection occurring. What we don't really understand to be able to really correctly answer that question is we don't understand impact from the ecological perspective. These trees are pollinated by generalists, so if we change these pollination or the flowering patterns, is that going to interfere with the pollination process? Also, I guess if with only 25% of those individuals showing some level of tolerance or limited level of impact, how much does that narrow the genetic diversity within this particular species? This is just some images just to show you the differences, what we're seeing. So these are individuals where we didn't see much in the way of dieback, absolutely covered in flower, versus those where we have significant levels of impact. Yes, you can see flower buds occurring on those, but it's a limited number in comparison to those that don't have any evidence of dieback. And it's just again showing that dieback. What we did notice, and we didn't actually capture a lot of this data, was that the buds appear to be quite small in comparison to those buds on the unaffected trees. The unique opportunity would have been to actually study the flowering patterns, the pollinator visitation rates on the different trees, but obviously we just haven't had the capacity to do that. But that's maybe something to look at for the future. The other species, and this is a key species, it's a keystone species ecologically, it's the broadleafed paperbark. This species grows in coastal eastern Australia from Botany Bay in New South Wales to Cape York in North Queensland. Again, similar to Melaleuca nodosa, it regenerates post-fire through resprout and seedlings. This is just to give you an idea of the different ecosystems in which it grows and which we assessed. You can see the different stand types in the top left there. We've got small diameter to stands, but they're almost in monocultural system. Here we've got a more inland woodland swamp ecosystem where we've got a range of different stem diameters, but again, it's almost a monoculture. In the centre, we've got here on the riverbank systems where it's a mix of different species that it's growing within. And then we have these systems where it's just pure monoculture because these areas are periodically inundated with water. We get these large, old Melaleuca quinquenervia growing in these sites. So in all these sites, we've seen infection occurring on the reshoots. First, when we started looking at these sites, they were getting affected primarily by insects. But once the insect damage had passed, we had that next lot of regeneration. Rust came in pretty quickly and affected both the foliage and the stems of the juvenile flush as it emerged. As a result of that infection, we've seen quite significant levels of dieback of the epicormic reshoots. And you can see the images here showing the different types of this is the small diameter tree on the left, and then we get these mature big old stands showing significant dieback as well. So as I said, we had a range of different sites where we looked at this. All sites except for one had difference in fire intensity. Our most northern site was on Gari or Fraser Island, where we had 71% of plants showing susceptibility. And you can see in all sites except for the Great Sandy National Park, where we did have that range in fire intensities, that we've got quite significant levels of susceptibility within these sites. Again, in the Great Sandy, we had very few individuals showing evidence of dieback at that point in time when we did the surveys. Remember, this data is from one-off surveys rather than looking at the long-term monitoring plots. The lower percentage that we see in the Gari site is probably the fact that when we arrived at this site, the rust was really just starting to get going on the new growth. So it's likely if we go back and revisit this site, that we'll see quite significant levels of dieback based on that susceptibility level. This is just looking at data from monitoring plots that we've established. Again, this is one of the sites we've got in Bundjalung National Park, where we can see again those links with the flush activity. So we get that build-up of disease following that availability of flush. After rust has killed off a lot of that availability of flush, we see a drop in disease and then a rise again as we get an increase in flush. What's interesting now is that we are actually starting to see deaths of reshoots, not just the dieback that's occurring, but deaths of these epicormic reshoots. And you can see in those images, particularly this one here, where we've got a high percentage of those reshoots dying off. So we haven't recorded too many actual tree deaths at this point in time, but I don't think it'll be too long before we start to see deaths occurring. This just to give you an idea of the rate of decline that we're seeing. The picture on the left is just showing that new flush covered in rust, and that was in May. Then we get that slow decline and it was burning off of all that new growth. We're looking at July there, and then that final image there is around August. And that tree now in that last assessment has probably, from memory, 50 to 60% of the reshoot that's completely dead on that tree. I'm not going to go into detail with seedling. We have captured some of the seedling data. This is just a couple of images really to show you what's happening with Melaleuca quinquenervia seedlings within these ecosystems. I think the one on the left is quite interesting in that it shows an infected Melaleuca quinquenervia getting killed off at the top, and all these other Myrtaceae that have had either no infection or low susceptibility now going to outcompete that Melaleuca quinquenervia for that space. And similarly, we've got in the other shots showing that significant levels of dieback, and other species such as the Leptospermum polygallifolium coming up around those infected plants. So this is the third species I want to focus on is Rhodamnia rubescens, or scrub turpentine. This species was considered common in New South Wales and southeast Queensland until rust arrived in 2010. And really before any evidence of fire damage, we saw significant reductions in population, so much so that it was listed as critically endangered, primarily in New South Wales, but I believe that's been done at a national level as well. So it produces resprout post-fire regeneration. We actually haven't seen any seedling regeneration since the arrival of rust, regardless of fire conditions. So what we do is see epicormic growth on the branches, the stems, but we also see root suckers occurring. And what we're really trying to see in this particular species is whether fire in combination with rust is going to accelerate the decline of this particular species and pushing it closer to extinction. So we've looked at 96 trees across 25 different sites. All these sites were in the Gondwana World Heritage Rainforest area. Of all those trees that we've looked at, 80% of those with the susceptible flush present showed some level of infection. It did vary depending on the site we went to, but there has been variations in obviously climatic conditions at each of these sites. Alarmingly, 70% of those trees had some level of dieback, some more significant than others. So this shows you just some of the images where we're getting relatively clean individuals where they've resprouted from the base post-fire. Unfortunately, in each one of these conditions, there is evidence of low levels of rust at the time of assessment, so it's likely that if we go back to these sites, we'll see significant levels of infection, knowing the susceptibility of this particular species. This just shows you some of the more severe damage we saw on the sites. So we've seen total dieback of those epicormic reshoots, in heavy infection and dieback of the root suckers, and dieback even on some of these large diameter individuals within some of these sites. Now, I just want to touch on eucalypt species. Now, previously we've not seen much in the way of rust infection in eucalypts. Angus Carnegie has previously reported low levels in plantations, but this is the first time we've seen massive regeneration occurring at once. And we did see high levels of infection occurring on some individuals. In particular, the blackbutt Eucalyptus pilearis, Eucalyptus planchoniana, Eucalyptus tyndallia, and the Eucalyptus amplifolia subspecies amplifolia. We did see also low levels of infection on the Crimbia intermedia and Crimbia henryi. And what was interesting with the Crimbia henryi is that it was getting so heavily impacted by the native fungus Quamularia pittorica that I think that was probably not allowing rust to come in and actually cause infection, because we know this species from our glasshouse studies is quite susceptible. In the images you can see there the infections we were seeing on seedlings, so quite small seedlings just emerging, the top one being Eucalyptus planchoniana, and then quite significant dieback on that bottom one there, which is Eucalyptus pilearis. This just shows you what we're seeing on the reshoots that were occurring. This is on the blackbutt Eucalyptus pilearis. Interesting, what we did see here, and I know this reflects a little bit of what's been reported in Brazil previously, is that we did see a canopy position effect, where the lower level regeneration was more affected than the mid-level, and then we hardly saw any infection in the upper canopy if regeneration was occurring there. And here it just shows particularly the middle photo and the one on the right, the middle photo being one that's had low levels of infection or no infection, versus one where we've had quite significant dieback as a result of repeated infection caused by rust. And this is just again reiterating what we've seen with seedlings, so a range of infection levels and a range of severity, and we have got monitoring plots looking at the impact of on survival of these particular seedling species. Again, this is just showing that dieback as well. This is just that shot of Carimbia, and just gives you on the spotted gum evidence that the Quambleria pittorica is causing quite significant impact. Very similar symptoms to what you would see with rust dieback. So thank you very much for that. I just want to thank my co-authors, Louise Shuey, Peter Entwistle, Rob Price, Fiona Giblin, Ryan Sims, the Butcher Land and Sea Rangers, who helped us out in Gari. And have been training additional people in identifying rust and helping us detect rust in different locations. Craig Steen and Angus Carnegie. Of course, the New South Wales Parks and Wildlife for their assistance, and the Queensland Parks and Wildlife and Department of Environment and Science for access to all these different areas. I particularly want to thank the funding bodies. This is multiple groups that little projects we've put together from funding sources, the Plant Biosecurity Science Foundation, the Threatened Species Recovery Hub in the Australian Network for Plant Conservation, and the New South Wales Department of Planning, Industry and Environment. Thank you.
Thank you very much, Geoff, and well done on getting through quite a spectacular number of slides. In about 25 minutes, I have 3 questions here. I think I'll just ask them in the order in which they were asked, and I think that's probably about all we'll have time for. First up, were the hosts, or perhaps a way to put it would be, to what extent were the hosts you talked about known to be highly susceptible before last year's fires?
Some of them definitely were. We knew obviously the Melaleuca nodosa, Melaleuca quinquenervia, and the Rhodamnia rubescens we knew were quite susceptible. Melaleuca squamea, we've never seen infection on that before. And just trying to remember some of those other ones that we had there. The Leptospermum speciosum was another one that we haven't seen significant levels of impact. But you know, we didn't necessarily try to target species we knew susceptible. We've got a lot of hosts that we've recorded that haven't shown evidence of infection, and they previously have not known to be susceptible. So the fire issue hasn't changed the way in terms they respond to rust. So.
All right, the next question. From what I could see, there were no pustules on the dieback branches. Is that correct? And if so, does that mean most of the dieback was due to girding rather than tissue infection?
No, that probably didn't show up on the images, but yes, on a lot of those we do see, and I assume you're talking about more so the Melaleuca quinquenervia. There was quite significant stem infection occurring on those. Again, those ones on the Eucalyptus pileolaris where we saw that significant dieback, stem infection occurred as well as on the foliage.
Very good. And does the abundance of rust differ with location of epicormic growth on the tree being higher for ground shoots and lower higher up?
Whether rust levels vary or whether we're just getting differences in infection as a result of that microclimate activity. We haven't actually looked at the spore levels, but we definitely saw that within the eucalypts, and we're trying to study now the Melaleuca quinquenervia, particularly those really tall trees. They, interestingly, the more mature trees and those taller trees have taken longer to reshoot after fire, and we're starting to only really see that regeneration occurring now. I guess we've gone into a period now where rust is generally lower, so we're probably going to have to wait till early next year when we start to get those peaks of rust level to really see whether there is a big difference with the different species in relation to that height of the epicormic regrowth.
Okay, and I see we have one sneaky question. It's definitely going to have to be the last question now. Any plan to try to measure inoculum levels?
No, not really within the scope of this. Certainly, it would be of interest to have a look in the different sites, particularly when in relation to the density of Myrtaceae within the different sites. Obviously, if you've got a whole area of high susceptibility or highly susceptible species on mass in one site, the likelihood that you're going to get higher levels of rust incidence in relation to mixed forests where Myrtaceae aren't dominant, it's quite likely we will get differences in spore concentrations.
All right. The lovely Robin Simcock has asked one more question, but I'm going to wrap it up now because that is all we have time for today, and a video of today's webinar answers to questions will be made available on the Beyond Myrtle Rust website in the next week. Just like to give a plug for the next month's webinar. That's also going to come to us from Australia. At the same time, Wednesday, December 9th, it will be given by Dr. Perry Tobias from the University of Sydney. She will be talking about the whole genome sequence for Ostropyxina obsidii and the further work this sequence enables, which is taking place in our Beyond Myrtle Rust program, as well as many other research efforts in Australasia and no doubt beyond. The recent publication of the sequence was achieved by a fantastic trans-Tasman effort involving many scientists from Australia and New Zealand, and I'm looking forward to hearing about that. We will see you all then. And Hannah, I realised I'm not quite sure how to kill this thing, so you might have to jump in, and I'll see you guys next month.